Welcome back to EASD TV. Now, the prize lectures are the jewels of the EASD programme, and the Claude Bernard lecture is the shiniest jewel of all. We are celebrating the 55th Claude Bernard lecture, and with me is the person who has won this prestigious award, someone who will be very well known to many in the diabetes community, Åke Lernmark from the Lund University in Sweden. Hello, and congratulations.
Thank you so much.
And I'm going to say something probably outrageous, but what took you so long? You've been such a distinguished and researcher in diabetes. I'm astonished you weren't lecturer many, many years ago.
I don't know, maybe the people who are, selecting the winners wanted more clear-cut results and outcome, and in type 1 diabetes for so many years there's a lot we did not know, and I would think that sort of colored the evaluation committee, saying they do antibodies, they do HLA, but they don't know how the disease develops. And
you are modest as ever after something like nearly over 600 papers, peer-reviewed papers that you've published, which is an extraordinary record for anyone. But tell me, how did you first get into this area of research?
Yeah, so this was serendipitous, and I was starting medical school in Umeå, University of Umeå, in northern Sweden. And the good news for me was that the school was brand new and everybody was needed. And on the train to Umeå from Stockholm, where I grew up and where I came from, I met a medical student who had a box with fat, obese mice with him. And he explained to me what he was going to do with research with these obese mice, and they had diabetes. And that's really the first time I heard about diabetes. And then I happened to end up in his department, and since everybody was needed at the time in the late 1960s to build up a new university, I was asked to help with teaching material. And then somebody asked me to measure blood glucose on these obese mice. So I said, okay.
You were hooked. You were hooked.
I was hooked, absolutely.
And where did your research take you then?
So my research took me, we had an unusual opportunity to do our own laboratory work at the university because everyone was needed. The professor usually came from somewhere else, Uppsala. So they were away and we were left alone to develop what we wanted to accomplish. So I got into insulin secretion by a British postdoctoral fellow who taught me how to measure insulin. And so in this way I did experiments where we were asking questions about cells in the islets of Langerhans, and people didn't know what they were doing. We know the beta cells make insulin, we know the glucagon cells, but there were two other cell types, and people didn't know what they were doing. So then I started to look at the possibility to find out. There's a cell type called D cells, and nobody knows what the D cell was doing. Why are they there? And it was very interesting that we were doing things and there was no answer anywhere. We had to find out ourselves.
So this is really curiosity-driven research at its very best.
Curiosity and asking questions. And then the real stepping stone for me was to meet Don Steiner, who discovered proinsulin. I think that's one of the Nobel in diabetes, one of the Nobel Prize winners that should have got the prize. He's passed away now, but the discovery of proinsulin was a major discovery. And he convinced me and my wife to come to Chicago. I met him, he became an honorary doctor at Umeå University, and I spent a whole night with him. You know, it never gets dark in Umeå during the summer, so we were talking until 5 in the morning, and then I got a 4-page letter from him arguing why I should come to his lab in Chicago. And that was a real eye-opener to meet very prominent scientists at the University of Chicago. So that's a long answer to your question how I got into
what
science.
What a wonderful memory. What a wonderful sort of idea it is to think of you, the two of you, all through the night, but in the daylight.
Yes, in daylight, that's right. And somehow we clicked. I was young and curious, and he was very experienced, very, very good scientist. And then he introduced me to other scientists at the University of Chicago. And I can tell you one of the fascinating things that you run into, the first nuclear reaction was done at the University of Chicago by Enrico Fermi. And he had his laboratory was across the street where I was working. And I needed a machine, a shaker, to isolate the islets of Langerhans. And I was recommended to go and talk to the physicist machine shop. And here is an old man. I talked to him, I explained what I needed, and he made a machine, and then I found out he was the laboratory assistant to Enrico Fermi.
Fantastic.
Unbelievable.
So what came out of your years in Chicago? Where did you then go in your research?
Yes, so then the major finding was that we had, from a pediatrician in Kansas City who had collected blood samples in children with newly diagnosed diabetes, I found that these children had antibodies which were binding to the beta cells. And they were binding to the surface, so they were called we called them islet cell surface antibodies. And I was working with biochemists, so that led to the question, what is antibody? We know the antibodies reacted with beta cells, but that's a phenomenon. It's not the answer. The question that I asked was what the antibodies react with. We need to find out, children have these antibodies and they shouldn't be there. So we were starting to do experiments and to find out what the antigen was. And that led, my 9 years of fellowship from the Swedish government was up, and I was recruited to a diabetes hospital in Copenhagen, the Steno Memorial Hospital, and was given the opportunity to start a laboratory called the Hagedorn Research Laboratory. And I had NIH money with me, and I was able to hire people, and I hired some very, very good people who continue the laboratory work with the identification of the antigen. And we found a protein that was known as the 64K protein, the $64,000 question. And we wanted, that was not enough. That was just an immunoprecipitated protein. And we wanted to know, what is it? And that took 10 years before we solved that problem through the work of my former postdoc, who went on with her own, Steinar Bekkeskov, with her own career. And I had a Danish postdoc in Seattle, where I moved to after Denmark. And he found this new GAD65 that was not described before. So that was pretty exciting.
So you've steadily been rewriting the textbooks?
Yes, in a way, yes. And so one thing, then we at least have a protein to work with. And that was as important to me and to us as the discovery of proinsulin by Don Steiner. He worked with proinsulin and the mechanism of making insulin. It's the groundwork to make recombinant insulin that the pharmaceutical company are producing today. It's really the groundwork. And so now we had a way to ask the question why the immune system is attacking GAD65.
There are so many highlights in your career. That was clearly a particular one for you.
That was so exciting, yes.
And can you recall the moment that you thought, oh my goodness?
Yes, I can. And we had a gene which is called the complementary DNA gene. And then upstairs at the University of Washington in Seattle, there was a group who were mapping all human genes to our different chromosomes. And GAD, the enzyme, was known to be on chromosome 2. So I went upstairs, talked to a colleague who was, the only thing she was doing, the Dr. Distech, was to take genes, put them on human chromosomes, and did radio autography to find out where is the gene binding. And then I know when she was going to be done, so I ran upstairs and had to ask her, what did you find? And she said, it's curious because we thought the gene would be in chromosome 2, but there's nothing there. Everything is on chromosome 10. So then I know we had something really exciting. It was a new gene.
Fantastic. That was a big highlight. Anything else that you particularly recall? Well, and there's many things, but that you particularly recall as highlights of your career?
So when I was director of the Hagedorn Research Laboratory in Copenhagen, we got I had my own funding. I had the funding from the foundation, and so I could bring in people. So we recruited 2 American molecular biologists who had just learned to work with cloning. And so then we were interested in the HLA system, why children with diabetes have to have a certain HLA type. And before it was all done by antibodies and serological typing techniques. So now, in collaboration with a group in Uppsala who had cloned the first HLA genes, we collaborated with them, got a probe, and then we found that the probe that connected to diabetes was not what we expected, was another one that people haven't seen before. So DR was known, but what we found was something else. And that's how DQ was cloned and detected, a gene that people suspected it should be there because of antibodies, but no one had seen it before. So that was equally exciting when we found out that we had come across a gene that connected to diabetes in a way that the other DR typing had done. So we know we were onto something important. That was exciting.
You have as much curiosity now as you had as a 20-year-old. For you, what are the unanswered questions that, had you another 50-year career, you'd like to tackle?
Yeah, so that is really to understand why a common virus infection in small children with the required HLA type, why that is ending up in poor virus antibodies, and the child is unable to clear the virus infection quickly. Why is that leading to an autoimmune reaction to insulin. You know, what's the connection there of being infected, being born with the risk genes, and then experience that your own immune system is attacking insulin or GAD65, you know, which are the two major antigens that we are working with. And it's really, we understand the mechanism how antigens, virus antigens are presented on proteins called HLA class 2. And we thought it would be simple that maybe there is an insulin-like molecule in the virus, or maybe there is a GAD-like component in the virus that is starting the self-reactive development, the self-reactive reaction, leading to a chronic, lifelong attack on these antigens. Nobody has an answer to that. And that's what I'm not, you know, I need to know. We'll do whatever we can, and we are working on it to find out how this really works. You know, it's hard to let this go. Why I want to know.
We're talking in the week that the Nobel Prize for Physiology or Medicine has been awarded. There are some prizes out there for research in diabetes, I'm sure, for the future. Where do you see those coming from?
I see in type 2 it will be to explain and have preventative measures of the pandemic of obesity associated with the development of diabetes. And I think all the different interventions and different treatments, etc., is not the answer. The answer is to find out what are the major risk factors that leads to this. And it's going to be details in terms of genetic risk, etc. I think the understanding on how the environment, what we eat, is influencing the development of the disease is going to be a major riddle to solve. That's type 2. In type 1, I think to find out the mechanism of autoimmunity through type 1 diabetes. I hope that type 1 diabetes is the leader of the pack to really clarify why an infectious disease, be it multiple sclerosis and Epstein-Barr virus, and other, you know, the rotavirus, the enterovirus for celiac disease. So now we understand the relationship, but we don't understand the mechanism, you know, what's really happening there. And we should be able to find out. I think that's the Nobel Prize.
It's been such a pleasure talking to you. Thank you so much. And congratulations, not just on the Claude Bernard, but this lifetime of work and curiosity that you've given us all. Thank you.
Thank you.
Well, what an interesting conversation that was. And early career researchers out there, Åke Lernmark has just outlined for you a program for the next 50 years of your research career. We'll be back soon. Bye for now.